In this presentation, I will be talking about the different ways in which mammals can be cloned artificially. Now, so far we've looked at natural cloning methods. In plants, for instance, we've looked at bulbs, tubers and runners, and also how plants can be artificially cloned using cuttings. We've also looked briefly at animal asexual reproduction in some simple animals like aphids and hydra, and also the cloning that takes place naturally when twins or triplets are produced and they are genetically identical to each other. But now we're going to look at the two artificial methods that can be used to clone mammals. Okay, firstly, you've got embryo splitting. So imagine you have a prize cow and a prize bull, but if they were just to reproduce naturally, you'd only be able to produce one or possibly two calves at a time, probably only one. So instead what they do is they extract the eggs using IVF techniques. They will also extract some semen from a bull, and what they would do is produce the embryos IVF. They'd produce them in vitro, and this would mean that they'd have these embryos, they could grow up these embryos, and then once they've grown up into a ball of cells, actually split them into lots of different balls and then implant them into different host mothers, not into the original mother. So this means you can produce lots of offspring from your two best parents. So this is a way of cloning that can be used to produce large quantities of meat and things like this. Now, the other method is the one that is to do with Dolly the sheep. This is using differentiated cells. So by somatic cell, we mean a body cell, not a gamete. And what happens here is the body cell contains all the genetic information for that a particular individual, and that entire nucleus is transferred into an egg, and then you clone the parent in this way. So these are the two methods. I'll just go through. So the cloning technique for somatic cell transfer, so this is the second one I discussed. So the egg cells are removed from the ovaries of a sheep, and then the somatic cell, so this is the cell containing all of the DNA, is removed from the udder of a different sheep. So this is what they did with Dolly the sheep. And then the udder cells, they actually grow them in these sort of starvation conditions. And what this does is it removes a lot of those epigenetic markers and means that they're no longer fully differentiated. Those markers that have been added that switch off a lot of that DNA are removed. And then the nucleus is actually removed from an egg cell. We say it's been enucleated. And the nucleus from that starved udder cell is injected into the empty egg cell. It's stimulated to divide by having a little electric shock, a little sort of depolarization. And then the embryo is inserted into the host sheep's uterus and grows. And this is exactly what happened with Dolly the sheep. Okay, now, cloning by embryo splitting is different. Cloning by embryo splitting, so for instance, they might have a ram and a ewe with very desirable characteristics, and the sperm and the egg are extracted using IVF techniques. And then an embryo is created in vitro, so it's grown up in the lab in a dish and is allowed to grow to the 16-cell stage. And then it can be split into more groups of cells, which can then be implanted into the uteruses of one or more host mothers. So this little diagram shows that. So we've got embryos are transferred to an unrelated surrogate mother. And this is just splitting one embryo and creating these identical twins or triplets. So you can see the two processes have different uses and different benefits. The cloning by, where are we? Oh, the cloning by embryo splitting is useful for creating lots of identical offspring. So this one is a more established method. It's less likely to go wrong. It's producing normal offspring that just happen to be identical twins, triplets, and so on. Whereas the nuclear transfer method, the the idea of that was to be able to use this method to actually clone from just one parent, maybe when sexual reproduction is not possible. So this was used to maybe clone organisms that can't reproduce for whatever reason. And it can also be used as a source of stem cells. So what they think is they could produce maybe a cloned embryo from a person who needed a liver transplant. And then instead of growing up an offspring from that cloned embryo, they could just use it as a source of stem cells. So they're the different reasons why you might use the two different techniques,